Okay, it's my pleasure here to invite our last speaker, Dr. Chris Austin, who's Director of NCATS at NIH. Dr. Austin, actually it's a similar parallel to what we just had from Nick, undergraduate at Princeton and his medical degree at Harvard. After his residency training in internal medicine and neurology, he ended up taking a job with Merck, and I think he got some of the sort of high throughput way of thinking of doing science, and then he brought that back to NIH in their chemical genomic screening program. He was a very instrumental player in the Tox21 initiative, which was the idea of like, let's use some of those approaches from high throughput screening for pharmacology to look at adverse events from the toxicology side. And so he's been a kind of, I think, a really key supporter of looking how adverse events and environmental factors can influence health. And now, as director of NCATS, he oversees all the CTSAs, the clinical and translational organizations across the country. And so I think that he has some very nice insight into how we're dealing with human disease and things. And we asked him to kind of share some of his experiences from his past time at NIH. So with that, Dr. Austin.
Oh, thanks. Well, it's great to be here, and my challenge is going to be what of the about 300 things I'd like to tell you, I actually will tell you, because there's so many thoughts that I have. But I have to say, before we get started, that though I know Eberhard and Nick didn't plan this, and we certainly didn't, because I never met either of them before today, it's interesting for me to see how what I'm going to tell you will reflect some of the things that they've said. The first, and I think at least Eberhard and Gary and I will share the interest in that knee in the lifespan curve that Nick showed, where he showed that the likelihood of living, goes like this until you hit about 50, 55, and then so we're in the stage of that. So I'm really interested in temporal scaling now. The other is what Eberhard said about learning math. Probably the best advice I was ever given by the first person I worked for ever who was a worm guy, C. elegans guy, and he said, if you ever have a chance to learn any more of any one particular subject, make it math, because what separates the truly great people from the not so great people is their ability to quantify, and certainly I've found that true over and over again. And you saw it in Nick's talk. The other thing that has made this even more important than it was is another thing that Nick mentioned, which I've also found in my career, which is that it's becoming increasingly easy for individuals to generate truly god-awful amounts of data. You heard Nick say that he didn't want to spend 7 years and 10 people counting worms or whatever it was. I actually did that as a postdoc. And in this case, we went from worms to cells, but it was very much the same. I spent 5 years in a dark microscope room counting cells with hand counters. And at the center I run now, I could have done my entire postdoctoral fellowship in a week. A week, no kidding. Which is lucky I'm doing what I'm doing now, because it's completely obsolescent. But what that means is that you simply cannot do analyses of these kinds of data sets in your head. You have to have mathematics and modeling in a way that we probably didn't need, back in the cottage industry days. When I was in training. The other thing that my other thought before we get going here is the thing I'll get to say multiple times, which is relevant, of course, to the exposome, is that we've moved from, at least some of us have moved from the single molecule explains everything phase to I think at least some of us are more willing to admit, frankly, our ignorance, and that we have to let Mother Nature tell us what the rules are, And you just saw this. I mean, Nick talks a great example of this. You know, he didn't start out with, I think this is important, and I'm going to cherry pick experiments to support my hypothesis. That's very popular, as you all know. Rather, to create a system which lets the data tell you what the rules are. And those of you who are fans of genetics will know this happened in the genetics world. Before the Genome Project, there were things called candidate gene studies, which I know the older folks will remember. And people would say, well, you know, I'm an expert in XYZ, therefore I know this gene is a candidate for that disease. And they were almost always wrong. And so I think people have they're more appreciative of our level of ignorance in biology and medicine. And the reason I tell you these parables is that I think it really does have relevance for the Exposome Project, and I'll try to get that across. So I'm going to leave you these slides so you'll have these. And you know, being at NIH, one of the things that's great about being at NIH is I was just talking to Judy Friedovich, we haven't seen each other in 100 years since we went to college together, who's on the faculty here, which is wonderful. So we just caught up on the last 35 years of our lives in the last 5 minutes. And one of the things that I was telling Judy is that, you know, one of the things that's wonderful about being at NIH is you feel like you're sort of in the middle of this grand central station of institutes and fields and even countries coming and going. And so I've played a large part in some of these and a small part in others, but they're all projects which you can learn from if you're trying to do a big biology project. And I'm going to go through some of these, not all of them. The first, everything down to here are things that are either positive or ongoing and look to be positive. The last two are cautionary tales, because as we all know in science, you learn perhaps even more from failed experiments than from successful ones, and so you want to look at these as well. Okay, so for those of you who were not born when the Human Genome Project was started, which probably, unfortunately, most of you, unfortunately for us who were on that knee of the death curve, it went from 1990 to 2003. The cool thing about this project is that it finished 2 years earlier and 10% under budget from what was projected. And so you really want to learn from this. I mean, this is, as far as I know, this is the only federally funded project which is ever finished under budget and under time. And rarely, will probably never be repeated, probably. But if you have an extreme phenotype like this, you want to study why. And so people have studied why this worked as well as it did. Some of it was certainly leadership. Jim Watson, who's at Cold Spring Harbor, where Matt Messelson is, and Francis Collins, of course. Importantly, there were 20 centers who all contributed to this. And this is something I've taken very seriously in virtually every project I've done since then, is this wonderful German adage, and I can't say the German, but it comes from a pathologist, a German pathologist in the 19th century, who said much is known, but unfortunately in different heads. And that sort of captures it. So you want to have as many heads as possible, but you also want to have real experts here. There's actually, of these, 25 of them actually contributed the majority of the sequence, and they're here. Another project, actually the first thing I did when I got to NIH was this knockout mouse project, which was, as you can see here, the purpose of this is to mount a high throughput international effort, so those two words will keep coming up, to produce and phenotype knockouts for all mouse genes and place these resources into the public domain. Now these principles are going to come back over and over and over again. High throughput, international, produce and phenotype all, the word all, and put in the public domain as rapidly as possible. And I should say, and if you can, diagram it in a way that makes intuitive, immediate sense to many politicians in this room, even congressmen. Because that's ultimately who you're going to have to talk to, or at least people who are not, they don't speak your lingua franca. And so we came up with this pyramid, was based on the mouse genome sequence, which was just completed at the time, and then making ES cells and mice, and then as we move up, the concept was that we would probably do less of this as we get to the top because it's more expensive as you go to the top. But that sort of helped crystallize what we're trying to do. The TOX program, and I'm going to go through just a couple of examples, and then I'm going to give you the distillation of some of the lessons. The TOX program, as you heard Gary mention, is I sometimes refer to as the mother of all systems biology experiments, because it really is. This project has as its goal to test 10,000 compounds at 15 concentrations in every pathway operant in mammalian cells over time, and then make all those data public. We've made about 100 million data points available public so far, this is one of the reasons we could do this is that it's a really great collaboration between the FDA, the EPA, people that I knew how to spell EPA, but I didn't know really what they did. I didn't even know they had an R&D group, but they do. It's a very big R&D group. Our group at NCATS and the National Toxicology Program, which is part of NIEHS. The goal of this project, and again, I use this slide all the time, is why I show you this, not so much to tell you about Tox 21, but to tell you how we explain this. Actually comes from a guy named Bob Kavlock, some of you may know, who used to be a big shot at EPA and R&D. And the idea was that organisms, ourselves included, are exposed to many chemicals. So when you go to the McDonald's for lunch and you have your double bacon cheeseburger, there are probably 1,000 potentially toxic chemicals that you put into your body, and they then achieve a certain tissue dose. And the way tox testing, at least in the environmental world, has traditionally been done, is that then you close your eyes and look for something called an apical endpoint. It's a term I'd never heard before I got into it, because I'm a genetics guy by training. That means something that the rat does that you can observe, gets a big tumor, keels over and dies, does that C. elegans thing where they just stop. So it's something that you can observe. But we never figured out what was in between. And so the next compound, you just do the same thing. And you didn't learn anything over time. And so the concept of this was, now that we know something about cells and networks and changes, phenotypes and molecular targets and pathways and all that, to take all those compounds and test them at all these different levels, sort of metaphorically taking the rat, dissecting the rat into its component cell types, its component pathways, testing all these chemicals on those pathways in those cell types, and then computationally putting the rat back together again. That's what we're doing in the project. And so we're trying to figure out what the pathways are that lead to toxicity or disease, because we don't know that. Eventually, this is the big kahuna, down at the bottom, of course, to be able to predict, develop predictive models. One of the things I learned when I went to Merck, much to my surprise, is that not only are we really bad at predicting what chemicals do, and small modifications of chemicals, which is why it's so hard to make drugs, but that we're really bad at predicting what biological effects they are. And I realized this when one of the adages when I was told when I first got to Merck was they said, you got to remember this. Every compound has two activities. You guys know this? So every compound, and I thought, okay, I'm going to write this down. Okay, go ahead. Every compound has two activities. The activity you know about and the activity you don't. Damn. But it's really true. There's always another one that'll get you. And that's why we're so bad at predicting. And you're not going to be able to guess. Sorry. It'd be nice if we knew enough, right? So you got to look broadly and let Mother Nature tell you. So that's what we're trying to do. But learn from the failed experiments too. So if you haven't read about the National Children's Study, I know Gary and others know a lot about this. This was a full-fledged fiasco, and there's lots of reasons for it. How that old saying about success has many fathers, but failure is an orphan. In this case, this has many fathers. There's many reasons for this, but like a phenotype, an extreme phenotype, which this really is, huge amount of money, a lot of really smart, committed people, all trying to do the right thing, and it just crashed and burned big time. And why is that? I think it's worth looking at it. We don't have time to go into it, but there have been a fair number of pretty good postmortems done here. So this, as you can see, it's always bad if you get on a scientific website and it says was a planned large-scale, blah 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 blah, authorized by Congress in 2000 and 2009, and then blah blah blah. However, decided to close this following advice of an expert review group. Basically, the decision was that this program looked like one of those. What was it? The thing? Where's Nick? Is he still here? Were those C. elegans when they move slowly? Are they yellow? Is that what they are? Before they turn red? Yeah. This was yellow. This was a yellow project. And then it was euthanized. Another one that was euthanized, that if you don't know, is it's an informatics example. And the best example the best description of this I could find was in Wikipedia. It's actually very well written, really nice references. I don't know who wrote this, but it's really well written. The idea, again, it was a great idea, and you can see this is a U.S. government program to develop. I'll read it better here. An open source, open access information network called the Cancer Biomedical Informatics Grid, and it was a fantastic idea. Again, crashed and burned. And so, as you're thinking about doing projects like this, take a look at these as well. Okay, so some of the lessons, and I'm going to jump around here and then get to a bunch of things to just think about at the end. So this is a great paper. Written by the three people who were responsible for most of the sequence being done. Francis Collins, who was head of the Genome Institute at the time, Michael Morgan, who was the head of the Wellcome Trust, which funded the Sanger Institute, and Ari Petrinos, who was the head of the Genomes to Life program at the Department of Energy. You all should know, if you don't remember, that it was actually DOE, not NIH, that started the Genome Project. So it's probably a little known fact, but it's really important to remember. Okay, and in this paper, there's a bunch of lessons, and they go through all this for you. But one is, don't try to go for the goal at the beginning. Stage what you're trying to do. Do one thing at a time. So there was huge effort put into these maps, genetic maps, et cetera. And it's a great term now. It's sort of obvious to all of us that if you're trying to go from here to California, or if I'm going from here to the airport, I'm not just going to get on the road and start driving. Say, how can I get there? Well, I actually have done that, and it's a bad idea. You really want to map it first, right? You're going to these days. You don't pull out a map. Does anybody know? You guys ever seen a map? But, I guess they still exist in museums and in my glove compartment. But but today, you Google map it, right? So you know where you're going. But you got to have a map. And so there was no map. People didn't even know how big the human genome was. The things like Cot curves that people used to do to figure these things out. So anyway, they came up with a genetic map, a physical map, then the DNA sequence, and then sequence variation, gene identification, and then functional analysis. And so it was very thoughtfully laid out. The other was to try hard to manage expectations, and this was an example of the argument that was made, at the time for why this is worth doing, and the argument was, well, compared to these really expensive things that have, perhaps a limited lifespan, this was projected to cost $3 billion. Completed, so it's projected to be finished in 2005. Again, this was 10% under here and 2 years under here, but the idea being that this is a gift that keeps on giving, and that was one of the reasons that Congress funded it in the first place. I must say that this really got out of hand as the Genome Project progressed, mainly because it came sort of a CNN-driven race to sequence the genome, between Francis and Craig Venter, and those two guys don't like each other, so they actually played into this. And so it created this expectation that we are still dealing with, that as soon as we had the genome, even though they didn't say that at the beginning, it was very clear. And if you read the National Research Council report that first proposed the Genome Project, it says it's very clear. But what happened was that it became the sort of common expectation was, as soon as we had the genome, all the scales would fall from our eyes, and we would understand all diseases instantaneously. And oh, by the way, because we understand them all, understanding is the same as treating, really. What's the difference? Wrong. That there will be treatments. So I had been at the Genome Institute, just to give you a sense of this, for about maybe 9 months. So I went to the Genome Institute to be the translation guy at the Genome Institute. So, the Genome Project is like chasing a car. The dog catches the car. What does it do with it? So my job was, what do we do with it? It was a great job, actually. So Francis is down testifying before Congress 2 weeks after the Genome Project got finished. And no lie, one of the congressmen asks him, "So, Dr. Collins, we're so excited to have you here today. It's been 2 weeks since the completion of the Genome Project, and we're so excited to have you tell us, what are the drugs that have come out since then?" And at that point, I thought, is there a return date on this job? This is just insane. Talk about expectation problems. So, so really, you're in any big project like this, you need to constantly balance getting people excited about what the potential is. And then as soon as they get excited, you say, "Okay, wait a minute, let's pull this back and, let's be realistic." And it's something that I've had to do over and over and over again. This concept of a community resource project, this is something that the Genome Institute came up with, or the Genome Project broadly, the people who did it came up with it, that I actually find pretty useful. In Talks 21, we've designated as officially a community resource project. And this is the definition from the so-called Fort Lauderdale meeting in 2003. And it's a project, this is quoted, a research project specifically devised and implemented to create a set of data, reagents, or other material whose primary utility is as a resource for the broad research community. So that's different from most. Most research project is, I need to publish my paper so I can finish my stupid PhD or get a job. That's the point of most, let's be honest. Can we speak freely? And we hope it'll be useful to somebody else, but there is a proximate endpoint. It's benefit me, not the community, or at least in that order. In this case, it's the other way around. And then the corollary for this is the community will be best served. This mission will be best served if the results of these projects are made immediately available to the research community. So you probably know that the Genome Project actually released data every 24 hours into public databases. There's a lot of issues associated with this, doing it with other kinds of data, but the theme, I think is worth thinking about. Okay, so how do you get something like Exposome done? It's been interesting. I've been talking with people about this since at least 2009. And it hasn't really caught on, and so it's interesting to think, well, why? Could be it's a bad idea, and it's a good reason it hasn't caught on. In this case, I don't think that's true. Could be that it's a good idea, but that its time has not come. So you got to wait for the technology to evolve. Maybe that was a little true, a few years ago. But just another anecdote I'll give you about that, which I think about all the time, is you can't wait for the technology. You got to start the project with the technology you have, and the project will drive the technology. When the Genome Project was first proposed in 1988, One of the hits on it was that with the technology available at the time, which was radioactive slab gels, remember those, it would take 130 years to do the sequence, to sequence the genome. Therefore, don't do it. Just wait till the technology evolves, and then we'll do it. And Watson said, no, that's a really dumb idea. No, we're going to start it, and we're going to have 30% of the project go to technology development, and necessity is the mother of invention. It'll drive the technology, not the other way around. And I think that's something that really applies here. However, fancy reports from high-level people also help. So both of this one, the Human Genome Project and this Tox21 project, benefited enormously from National Academy of Sciences reports, and these are quotes from each of these, which really have come true in both cases. But the important thing is that there were some really influential people who said this will be game-changing. So it's worth putting substantial resources into this, and to do it in a visionary way. And they really did this in both cases. Project papers can also be very helpful. So I showed you the mouse knockout project paper. This is the ENCODE project, if you know about that. That's all the structural and functional elements of the human genome. That was one of the things that was done after the genome project was finished. The HapMap project, which is it was the first project looking at inherited human variation across the genome. This Molecular Libraries Initiative, which I also ran at NIH, was another broad, big science project. What's interesting about these projects, about these papers, is that the papers in themselves create a reality, which is interesting. That we're all taught that what we ought to do is wait until we have the data and then write the paper to report on the data. And there's a lot of, for data-driven papers, of course, I hope that's what you're all doing. Otherwise, your mentors are in trouble. But in this case, if you're trying to capture the imagination of the scientific community, if you can write it as if, and then it takes on a life of its own in a way that otherwise it might not. And here's the one from Tox21. When you're in the middle of a project, it also helps to keep writing project updates, because these projects can take a long time. Genome project took 13 years. And so you can't wait till the end to write the paper, although that was, lots of paper came out at the end. But the Genome Project actually had 5-year plans. Every 5 years, they published another plan. And this happens to be, I just chose the one 1998 to 2003, but there are ones before and after this. And this is a mid-course paper that 4 of us wrote about the Tox21 program, sort of in mid-course, and this was from 2013, which also helps people appreciate what the project is actually producing. Something that's really important, I think, in all of these projects, but particularly in the exposome, is community engagement. Now, we as scientists are not very good at this. I must tell you that most of us, and I include myself in this, we tend to be a rather introverted lot. And so it's not. We're not people who are like Donald Trump out there, saying, we're taught to be very careful and considered in what we do, and we're also taught to have these really weird idiosyncratic languages, to use terms like the haplotype map. What the heck is the haplotype map? Can't you speak in English? So this whole idea that we would engage directly with people who are going to be affected by the research we do is a little scary. It's something that, frankly, I've really forced myself to do over the last 25 years, and it's added both enormous richness to my research, as well as made it possible to do projects which otherwise I think wouldn't have been possible. So the granddaddy of this is actually the ELSI program and the Genome Project. So something called the ELSI stands. It's not a cow. And ELSI is a cow, but not in this case. This is ethical, legal, social implications. ELSI, ELSI. And that was a project that well, actually by statute, 5% of the Human Genome Project budget went to ELSI. You know, considering, okay, when we have a human genome sequence, what does that do about privacy? Turned out to be. So Genome got ahead, the Genome Institute got ahead of that problem. Didn't wait for the genome, you know, have something terrible happen and then have to back up. So, and certainly, you know, for exposome, you see, it was a community engagement, public consultation, and sample collection groups. This was part of the project. Something that I think helps with all of these, that I was taught about by the former NIH director, Elias Sahuni, who was Nick, an engineer. In my opinion, that's one of the reasons he was good, because he thinks differently. He thinks about connections, not just about individual circuits, right? That the whole, or individual components, that the circuit has to work. You know, that's how engineers think, and you saw Nick's thinking here. Is to think about what's sometimes called a time risk matrix or time difficulty matrix. When Elias was, when we were proposing new projects at NIH, he required everybody to put them in this form. And so what is it? It's you put time on the x-axis here, 1 to 3, 4 to 7, 8 to 10, and increasing level of difficulty over here. And then what you do is you take, what do you want to, what's the ideal state? Where do you want to be? And you put that in the upper right corner up here. And then you methodically think backwards. Okay, in order to make this possible, what do I have to do now? At the very beginning, something that's relatively easy, that without having all of this as the foundation, there's no way I can build my beautiful building on top of it. And you actually write this down. And I've done these in great detail, of lists of things that we're going to do in 1 to 3, 4 to 7, 8 to 10, and then you had stretch goals in each of them. It's incredibly helpful. It's kind of like writing a grant application, but a 10-year gigantic grant application, because these are big science projects, but very, very much worth doing. Okay, so that's those are the sort of examples. So lessons. So what's the derived lessons? There's 3 pages of these, so stay tuned. Okay, so some is probably obvious, at least to most people. Some scientific questions are simply so complex, so large in scope, and or so inherently multidisciplinary that they simply have to take on this sort of big science approach. Now, the Genome Project, if you're trying to sell this, there's lots of other examples, but the Genome Project is still the one that everybody knows about, so that, and it's fair game to talk about that. Everybody still does. Secondly, large-scale projects often spawn entire new fields of investigation. It's probably for a lot of you, it's hard to imagine a world that didn't have large amounts of informatics in it, or bioinformatics. But to a great degree, the whole field of bioinformatics was created by the fact that large amounts of sequence data started getting put into GenBank, so that you couldn't do work requiring. Like when I was in training, we had these things called pile-up. You remember that? You know, where you could get these really basic platforms to compare sequences, because there were only a few thousand sequences in GenBank. And now there are billions, right? So you really need. So there was actually a whole field that was created as a result of this. As a result of the Molecular Libraries Initiative, the same thing happened in chemical informatics. So because all of a sudden there were millions of data points of chemicals in their biological effects. Same thing that's often said about try to sell NASA. You remember this? You know, where NASA, you should fund NASA because that's where Teflon came from. You probably heard that, and it's actually true. Secondly, or thirdly, big biology projects produce data, certainly, that answer large questions. That's why you're doing this. However, something that I continue to think I see is underestimated over and over and over and over again, and results in unnecessary conflict among otherwise perfectly reasonable scientists, are that these data are hypothesis generating, and they enable individual labs. I mean, think about the Genome Project. How many individual labs has that enabled to do work in a far more sophisticated way than they could do before. I mentioned this before, new technologies are frequently created and become smaller, faster, cheaper under pressure of the project. Necessity is the mother of invention. I hope you've all seen the comparison of genome sequencing cost versus Moore's Law, and they paralleled each other for a while, and in about 2003, when the original sequence got done, there was an effort to really increase the throughputs, and so the cost of sequencing diverged from Moore's Law dramatically. And so the cost of sequencing has gone down 10,000 folds in the last decade or so. It's just unprecedented. And it didn't happen by accident. It happened for a very clear reason. In most of these projects, I've found that you really need to divide this into data generation, technology development to develop new ways of generating the data in a better way, and then really focus on dissemination. Don't leave it up to, we'll publish a paper sometime. Really focus on this problem. And you may have to create entire new databases to do this. So this is an example, when we did molecular libraries, we had nowhere to put the data. So we created something called PubChem, probably a lot of you know about. That was created for the molecular libraries program because we had nowhere to put it. So we had to create a whole new part. It's actually one of the biggest parts of NCBI now as a result of that. And again, the ethical, legal, social component could be important and helpful, both to help the project move forward and for advocacy reasons, and also keep you out of trouble. Because these kinds of translational projects, you've probably seen this, that you can result in, these things can have train wrecks in the public eye that I think many of which are avoidable, but you have to think about them beginning. Second, really important, you have to assemble a team of people who want to play on a team. Not all scientists want to play on a team. Some of my best friends are marginally autistic. They don't want to talk to anybody. And that's fine. That's absolutely fine. There's just wonderful things that those folks do, and they're some of my closest friends. But you can't have them be on a big team. They're just uncomfortable. If you've taken the Myers-Briggs thing or any of those personality surveys, it just doesn't work. And we tend to think, well, we're just going to have scientific expertise. That's not sufficient. I mean, it's necessary, but not sufficient. So I always think about this like a basketball team, right? And if you're going to put together a basketball team, you can't have a bunch of people who are just going to hog the ball all the time, or they can't be the greatest free throw player in the world. They gotta want to play on a team. The thing that you will sometimes be told is team science is not real science because it devalues the importance of the individual. Give me a freaking break. That's like saying that Kobe Bryant is not a good basketball player because he plays on a team. It's ridiculous, but you'll hear that. And so just push back. Or have him call me. An active and scientifically accomplished project management group is really essential. Again, if you're going to have a team, you got to have a coach. You can't just put 11 players out on a football field and have them just sort of running around. They're going to look like one of Nick's stochastic things. People like the kindergartners in the playground. So project management, something I learned at Merck, really important. But these people have got to be scientific experts. They can't be people who couldn't do science, so they're in project management. And there are a lot of those people. So avoid those people like the plague. They have to be really good and interested in project management. It's a really hard job. Secondly, having a respected, involved, and this is really clear, people are really good at detecting figureheads. So respected, involved, and independent group of advisors is really important. Both people that you can go to and say, Eberhard, you're really full of it. I love you, but you're full of it. That's really some of the best thing I've ever been told are those things. And you need a group of internal advisors to do that. But also external advocacy, external validation, really important. Staging with measurable outcomes, we talked about that. Making data as freely available as possible. Consider this as a community research project. Helps maintain support of the community. And don't try to be all things to all people. One of the things that happened with the children's study, and you'll hear a lot around the postmortems that happened as a result of the demise of the Genome Project, the children's study, was that it became so popular that everybody wanted to hang their ornament on the children's study Christmas tree, and it collapsed of its own weight. So it needs to be big, but not too big, and you can't try to do everything. And you've seen in the new ECHO program that you've seen, which is an interesting term, given where that project came from. But it's that they're really trying to avoid this. They're trying to be big, but limited. Okay. Lastly, this is really hard, but it's really important. Project has to be driven by science, not by politics. This is really hard because big projects require large amounts of funding. Large amounts of funding inevitably get you into advocacy, right? I mean, NIH is a taxpayer-funded organization, and so a lot of you probably do advocacy on the Hill, or maybe you don't. I'd encourage you to do it. It's really it's an important thing to do for all kinds of reasons. But the important thing is that politicians know as much about science as scientists know about politics. But they will often try to get involved and manage the project. It's another reason why the children study failed. So try to, and one of the ways around this is, Congress is very often what they're focused on is stewardship of public funds. It's a really good thing. And so if you have an external board that they can look at and say, look, a group of independent, really smart people like the National Academy said this is a great idea, then Congress was comfortable that you're not wasting taxpayers' money. And so it's an important balance. Especially for talking to your colleagues who think that the exposome is some kind of new cleaning fluid, as well as congressmen, congresspeople, have the 2 to 3 sentence elevator speech. What the opportunity is, what are you trying to do, who's doing it, what's it going to deliver, and what bad is going to happen if we don't do it. So those 4 things, you got to remember those things. Now, lastly, you got to expect pushback because it's going to happen. So early on, the Genome Project was said to not only be impossible, because this could take 130 years to do it, but even if you did it, it'd be a phone book nobody's going to use. Who the heck wants a 3 billion letter Word file, right? It's a frickin' phone book. So, and this was said by a lot of smart people. Perspective expectation management is a must. We talked about that. Positively, proactively address this concern that's going to come up. It comes up over and over and over again. In a flat budget environment, to fund something really big like Exposome, there's going to be concern that individual investigator R1 grants or R3s or R whatevers are going to suffer, and so you have to be honest and address that problem up front. And there are ways to do it. Every project has dealt with this, but don't try to hide from it because it's going to happen. Lastly, the big science approach or big biology approach, it's different from how most science is done. Still these days, most science and most grant applications are hypothesis-driven, testing a hypothesis on a single molecule, single gene, single protein, or maybe a couple, small number, and that's what scientists, especially who were in the gray head stage, the knee of the death curve, that's the way they were trained. To some degree, because there was no way to do the all experiments, the comprehensive experiments. But you will also run into almost a moral objection on a part of a lot of our scientific colleagues who feel that if a project is not hypothesis-driven, it is what am I going to say? Fishing expedition, right? Yeah, thank you. And So you just have to get ready for that. And again, there are ways to explain that. I think it's a canard, but just realize that it's different, and you're going to have to educate your colleagues about this. I think one of the reasons that it makes people uncomfortable is that it makes scientists do what we most hate to do, which is admit that we don't know. And so you're making people uncomfortable by doing this, and just realize that's what you're going to do, and just plan for it up front. So I hope those were enough lessons to learn. I don't know how much time we have for discussion. Unfortunately, I have to get to the airport and get back for a meeting. But I'm glad to answer any questions, and hope to give you some food for thought for after I go. So, thank you.